morning, students. It's my pleasure to be presenting today the talk on bacterial membrane functions, which is more than just check it. Now, coming to the cell structure, in prokaryotic cells, the structures of the cell envelope vary depending on the type of cell and organism. Most, but not all, prokaryotic cells have a cell wall, but the makeup of this cell wall varies. The electron microscopy images of the gram-negative and the gram-positive cells show dense layers, electron-dense layers, which are separated by less dense space, the periplasm. In gram-negative, external to the periplasm is another electron-dense layer, which is the outer membrane. Thus, the gram-negative bacteria has 3 layers as compared to the gram-positive bacteria, which has 2 layers. Now, all cells, be it prokaryotes or the eukaryotic cells, they have plasma membrane, which is also called as the cytoplasmic membrane or the cell membrane. Now, the plasma membrane structure was first described by Singer and Nicholson as the fluid mosaic membrane model. This structure was based on the thermodynamic principles and the available data on component lateral mobility within the membrane plane. However, after a few years, more than 40 years, the model still remains relevant for describing the basic nanoscale structures of a variety of biological membranes. Nonetheless, an updated fluid mosaic membrane model, which was described by Jacobson and colleagues in 1995, they have emphasized on it replaced the mosaic nature of the cellular membranes, where the proteins and the lipid components are more crowded and limited to their movements in the membrane plane by lipid-lipid, protein-protein, lipid-protein interactions, as well as cell matrix, cell-cell and cytoskeletal interactions. These interactions are important in restraining the membrane components and maintaining the unique mosaic organization of the cell membranes into functional dynamic domains. It's difficult to portray in any of the cartoons the concept that the components of the cell membranes, which functions in various domains as non-uniform, non-random, cooperative and dynamic elements in thermodynamic equilibrium. So moving on with this understanding of the modified or the updated cell membrane model, we will delve a little bit more into the functions of the cytoplasmic membrane in bacteria. Like the cell membranes in eukaryotes, the cytoplasmic membranes in prokaryotes functions as permeability barrier. Further, it also serves as an anchor for membrane proteins, serving various functions that are crucial to the survival of the cell. Noteworthy among these are the presence of proteins and complexes assisting in energy conservation, which in case of eukaryotes happens in mitochondria. One would often wonder, how does these proteins get embedded in these membranes? And are there proteins in the periplasm as well? So proteins to be either embedded in the membrane or to be transported into the or secreted out into the periplasm are destined right from the beginning at the start point of the synthesis. In simple words, not venturing into the complexities and exceptions of the biological system, each protein to be secreted out contains a sequence of amino acids, the signal recognition sequence that is recognized by the cytoplasmic component of the translocase system. This cytoplasmic component binds to this or identifies the signal recognition sequence and binds to this protein through this signal recognition sequence, presenting it to the translocase for further action. Depending upon the requirement, the protein either gets embedded in the membrane or is exported into the periplasm. Now, upon getting embedded into the membrane, the proteins undergo folding depending upon with the help of the membrane-based chaperones and depending upon the nature of the work to be done. Proteins are exported through and inserted into prokaryotic membranes by activities of other proteins which are called as the translocases. A key one of these translocases is the sex system, standing for the secretory system. The sex system exports both proteins and inserts membrane integral proteins into this membrane. On the other hand, certain other proteins which needs to be folded into the periplasm in a pre-folded manner, those proteins are transported across the plasma membrane by the TAT system. The abbreviation TAT stands for twin arginine translocase, where the protein to be transported contains 2 arginine amino acid residues for the identification. The signal sequence on the folded TAT protein, is recognized by other components of the TAT translocase system, the TAT BC proteins, which carry it into the membrane transporter, the TAT-A protein. The whole process utilizes proton motif gradient as a source of energy. The proteins embedded in the cell membrane serve as permeability barrier, allowing transport of specific substrates across the membrane by utilizing energy. Depending upon the energy source, substrate transported and direction of transport, these proteins have been categorized into either simple transport proteins, group translocases, or APC transporters. Further, they are also been categorized into uniporters, antiporters, or symporters. The simple transport system, it is driven by the proton motif force and is involved in transport of substance, or the substance which is transported from the outer side to the inner side along with one H+ ion. The group translocation system chemically modifies the substrate transiently to be transported inside into the cell. This chemical modification of the transported substance is driven by phosphoenol pyruvate, which is a intermediate high energy component in the cell. Now, as the name suggests, group translocation works for a group of substrates, mostly a group of amino acids or a group of sugars. Group translocation systems for sugar transport such as glucose transport or fructose transport has been very well studied. The third type of protein which utilizes ATP for transport of substrates from the outside into the inner cytoplasm of the protein is the ABC transporter. As the name suggests, ABC stands for ATP binding cassette. The ABC comprises of 3 subunits, one being located in the periplasm, which is the periplasmic binding protein, and the other is the transmembrane domain, and the third one is the ATP hydrolysis domain. For transport of substrates through these ABC transporters, it involves utilization of ATP as the energy source. So, besides these basic proteins that we have already mentioned, the gram-negative bacteria utilizes a number of dedicated protein secretion systems to transport proteins across 1, 2 or 3 phospholipid membranes, as the case may be. Some proteins are secreted in a 2-step manner through the SEC or the TAT dependent mechanisms, as is shown over here. These proteins cross the inner membrane with the help of either the SEC or the TAT secretion pathways and are then transported across the outer membrane using a second secretion system. The type 2 secretion systems and the type 5 secretion system secretes protein in this manner. Because both of them secrete folded substrates. The type 2 secretion system translocates proteins initially either by the TAT or the SEC pathway, which we have discussed previously. In contrast to this, the type 5 secretion systems are autotransporters. These autotransporters, the proteins must be unfolded prior to the outer membrane transport, and thus must be secreted across the inner membrane by this SEC pathway. So, once again to reiterate, while the type 2 secretion system utilizes protein either from the SEC or the TAT pathway, the type 5 secretion system utilizes proteins which are secreted to the periplasm through the SEC pathway, because it is capable of secreting only the unfolded proteins. Additionally, several gram-negative protein secretion systems such as type 6, type 7, type 8, type 9 and 10 and 11 have been identified. These gram-negative protein secretion systems transport their substrates across both bacterial membranes in either one step, such as the SEC or TAT dependent process. Now, these include the type 1 secretion systems, which is shown over here, the type 3 secretion systems, the type 4 secretion system and the type 6 secretion system. All of these pathways contain the periplasmic spanning channels and secrete proteins from the cytoplasm to the outside of the cell. However, the mechanisms of protein secretion in each of them are quite different. Now, 3 of these secretion systems, the type 3 secretion system, the type 4 secretion system and type 6 secretion system, can also transport proteins across an additional host membrane, delivering the proteins directly into the cytosol of the target cell. So, as you can understand, or as you can imagine from this ability, so they are capable of delivering effector molecules directly from the bacterial cytoplasm to the host cell. As elucidated here, in addition to whatever the secretion systems we have mentioned previously or discussed previously, the gram-negative bacterial outer membrane consists of channels that allow in and outflow of nutrients in a non-specific manner. These are slightly different from those that are embedded in the cytoplasmic membrane, which are either the single component transporters or the multi-component transporters. The multi-component, so these proteins, they serve as the efflux channels, secreting the proteins which are present in the cytoplasm, in the periplasm region, to the outside of the cell. Now, these multi-component, some of them are multi-component efflux systems, and these multi-component efflux systems constitute of the outer membrane proteins, the periplasmic adapter proteins and the cytoplasmic protein to form a functional tripartite unit that is capable of extruding molecules from the cytoplasm as well as from the periplasm. This classic example of such an arrangement are the R&D efflux pumps, which has been extensively studied in drug resistance, as they extrude drugs from the periplasmic space to the outside, preventing their accumulation in the cytoplasm, thus lowering the cytoplasmic drug concentration and inhibiting killing of the organisms. Now, little bit more into this efflux pumps. Again, there is a great deal of diversity depending upon the the structure of these efflux pump proteins, the mechanism of their functions and the energy usage. So basically they are classified either into the MFS, SMR or the mate family, and these are mostly, they function as drug proton or cation antiporters, whereas the ABC transporters that we discussed earlier, these are also capable of effluxing the drugs and they use the energy from the ATP. Apart from that, you have the R&D efflux pumps, which is the tripartite pump that we discussed in the previous slides, and these are capable of effluxing the drugs directly into the outside environment, utilizing the proton motif gradients across the periplasmic space. Now, moving further from this, membrane proteins are also very important to sense external cues through, and most important amongst this group of proteins that sense external cues are the two component regulatory systems. Now, these two component regulatory system comprises of the membrane integrated sensor histidine kinase and the cytoplasmic response regulator. Now, bacteria respond to changing environments by altering the gene expression. The two component regulatory systems are important mediators of this signal transduction and enable bacteria to detect any physical and or chemical changes and then relay this signal through the cytoplasm to the bacterial nucleoid where modulation of this gene expression occurs. Upon sensing an external stimulus, the histidine kinase undergoes dephosphorylation, transferring the phosphate group to the response regulator, and this response regulator goes and binds, triggers downstream processing through signaling processing and triggers differential gene expression in the cell. So essentially, these proteins pass on the environmental cues through phosphorylation and dephosphorylation cascades. Coming to another important function in eukaryotes, the mitochondria, more specifically the inner membrane of the mitochondria, help maintain a proton gradient across the intermembrane space and the cytoplasm that drives the formation of ATP. A simplified illustration of this is shown over here, where the mitochondrial electric transport system proteins are labeled as 1, which is the NADH dehydrogenase, now 2, which is the cytochrome BC complex, 3, which is the cytochrome C oxidase complex, and the electron carriers over here are referred to as Q and the cytochromes is referred to as C. The conversion of ADP plus phosphate into ATP occurs at this complex 4 or the protein channel 4, which is also the ATP synthetase, which we refer to as the F1 F0 particle. Now this ATP synthase moves hydrogen ions back into the matrix where they are again used by these sites as 1, 2 and 3 mentioned over here. These carrier mediated diffusion exchanges of the ATP and ATP between the matrix and the inner membrane space occurs on a regular basis, and from the inner membrane space to the cytoplasm of the cell or the cytosol of the cell, these ATP molecules are pumped out through again outer membrane transport proteins. Now, if you compare this structure of the mitochondrion, it resembles to that a great deal to that of the bacterial cell membrane, gram-negative bacterial cell membrane, where you have an outer membrane, inner membrane separated by a periplasmic space. Indeed, in prokaryotes or in bacteria, this proton conservation of energy is accomplished by the cell membrane. The bacterial cell membrane, also known as the inner membrane, is the site for electron transport proteins and ATP synthesis. The electron transport protein chains help generate a proton motif gradient which aids in accomplishing multiple functions, including ATP conservation or the energy conservation, flagellar rotation, solute and ion transport. Now, as mentioned previously, efflux pumps also utilize this proton motif gradient to expel substrates out of the cell. Thus, proton motif gradient is extremely crucial for survival of the bacterial cells. Besides transport, Proton motive force also helps accomplish multitude of functions in a bacterial cell, including but not restricted to flagellar movement, including but not restricted to pH homeostasis, motility, as I mentioned previously, ATP synthesis, membrane transport, as well as the dynamic communication. So, the bacteria also utilize their membrane potential as a means of signaling and processing information. By depolarizing this membrane potential, bacteria enter into a non-dividing transient state, which is the persister state, wherein they are more resistant to antibiotics. Homeostasis of pH is also controlled by proton pumps, which maintain the proton motive force and thus are dependent on the membrane potential. Apart from this, cell division also requires membrane potential for proper localization of the cell division site. You all would have heard about the formation of the FTS Z ring, and this formation of the FTS Z ring is important or is driven by this proton motive force. Delocalization or disruption of these proton motive force leads to inhibition of cell division, and this can easily be studied by using native proton force. Lastly but not the least, the bacterial membrane potential can respond dynamically to the molecular and electrochemical signals, thus allowing for the intracellular communication. So one can very well appreciate the diverse roles of these membrane potential that it plays, and this would not have been possible without the presence of this membrane. Recent studies have also shown important function or important role of these membrane potential in mediating resistance towards antibiotics or tolerance towards antibiotics. Bacteria, by lowering the membrane potential, lower the overall physiology or the physiology of the cell, thus reducing the availability of targets in the cell, which leads to non-function or reduced efficacy or efficiency of the drugs to function. Two such studies which I have shown over here, there are many others due to constraint of the space and also intricacy of the understanding. I have just included snapshots of these two. So what over here in these papers that it mentions is that the cytochrome BD, as I showed previously, the cytochrome complexes which are involved in the electron transport chain, they help in regulating biofilm, antibiotic tolerance in biofilm, and help in regulating accumulation of obnoxious chemicals inside the cells. It has also been shown that not all bacteria, respond to this. All bacteria respond to this. There are certain bacteria where nitrite also modulates tolerance towards antibiotic. Now, nitrite, it does so by inhibiting the heme copper oxidase of the cytochrome in the bacteria. So students, by the end of this talk, you would have all appreciated that it's more than just check kits. So the membranes play a very important role in bacteria because they are the primary source not only of the barriers, but also they function they play a very important role in the day-to-day functions of the bacterial cells. So with this, I would like to thank the Bookworms and my team Microbes for giving for the Bookworms for giving me this invitation and their patience in the delay that happened while sharing of the slides, and my team Microbes who delved with me into the lives of this bacteria. Thank you.